you very much, Leila, for the introduction, but also first for the invitation to be able to actually present to you. And hopefully you also see my slides already. All right. So just to confirm that I don't go on for too long. And yeah, I mean, you probably read a little bit, you know, in some gloomy words, what I'm trying to do with you today, to go through multiscale bone biomechanics and imaging. There's a number of terms that are important in that and have been important in my career. And maybe it's also important in that sense, say, talk just a few words about, you know, myself in terms of not achievements, but where I come from. So I think you know, I'm an electrical engineer by training, and electrical engineering at the time when I did that, so this is the 80s of the last millennium. Long time ago. It was really an applied mathematician. So you basically studied mathematics, but with an application on it, right? So doing communications, for example, or, you know, that was the time when you know, wireless networks were invented and things like that. So you need all kinds of mathematics for that. But that wasn't so appealing to me. I'm a very visual person. So I thought, what about applying mathematics to detecting features in images and doing image processing. So also started interested to be using imaging and modalities, developing imaging modalities that would allow you and combine that with information systems, let's put it this way, right? So it's really like a very classical thing that you've seen, you know, in terms of the development that are going from communications and now we have some emergence, right? So like you have your mobile phone, let's say, your computers, all of these, they all, you know, digital, they all information technology, they all use image processing to identify you, your faces, all kinds of things. But of course, this kind of development is also shown in medical sciences if people are interested in imaging, image processing, and we're using much of the same technology. And of course, we can profit though from the mass markets and making things always faster and allowing us to have such capabilities now basically also on our phone. So I could probably run this whole meeting in terms of webinar on my phone, but of course I can't do all the research without still the supercomputers we are using. So it's just a little bit of an introduction that is on the side and on where it comes from. But today we have a team that is very interdisciplinary. We do all kinds of different work, and I will not talk about all of the parts. I'll talk about the longest streams of work so that you have a little bit of a historic perspective as well on where the work is coming from, especially on the imaging side. And so I'm not excluding some of the newer work we're doing in the more cellular and molecular biology of organoids, for example, or in vitro systems working for bone replacements and platforms to study bone in an in vitro system. So I will exclude that part. But let's go through the story in why bone maybe is important as well. So try to go ahead. So something that has been not just because I'm aging personally, it's of course something that has been accompanying me throughout my career, that I was always interested in the effects of aging and or people at older age, because these are the people, and especially women, that develop osteoporotic fractures. And so this is of course a hallmark as well, that, you know, that people with age they have, of course, approaching old age in unprecedented numbers, but the prevalence in age-related diseases, frailty, and the disability are visible signs of this demographic shift, which is really dramatically affecting the incidence and prevalence of osteoporotic fractures. So this has really been already at the beginning of my work. We were interested to have diagnostic tools to allow us to better separate people that would eventually fractures from people that are hopefully safe and will go on and don't need medication, for example. And so this was also developed when there wasn't any medications available, so you would be able to have more preventative approaches. And of course now there is a patient treatment is available, and it goes along though with imaging modalities, testing that gives you quantitative results on whether, for example, you are prone to fracture or you also basically responding to the therapy that you have. So, some improvements, or at least not getting worse. So this is the origin a little bit of the work we're doing. And so, the images on the side, you can see somehow some very old pairs of imaging they're quite famous. They've been published in a newspaper in USA Today, but that still is quite important. They were the first time in USA Today, was at the time an important newspaper in the US, that people were saying, like, this is what a young normal looks like. This is an osteoporotic person. And, there was a set of micro-CT images that we have done very early on in one of our studies. So it's important to show these, because that was the first time that you could do some kind of a histology in 3D, getting histological quality of the images, and also able to quantify those. So it's not, I will show a lot of beautiful pictures, I think are beautiful, but the important thing is that they are quantifiable. So you can get an idea of what's going on between the young normal and osteoporotic, but maybe it's not so easy actually to see that, and I will get to that, why biomechanics comes into that. So this is just a pure imaging approach, but you can maybe imply some biomechanics in that. Yes, it looks like the top one is stronger than the lower one, if you think about it, if you think a little bit about how it's built, but it's not so easy to see that. And so there is, of course, a large efforts still to develop also more novel technology, allowing 3D microstructural imaging of mechanical bone function. So not just the pure image, but also knowing about the mechanical function. How stiff is the material? You could do mechanical tests, but maybe you don't want to do that. You just want to get that non-destructively. So you could also use it in patients for better diagnosis and early fracture prediction. So the motivation that we have these days, although because diagnosing osteoporosis is actually a difficult task. So you're expecting the whole population, we were really specializing a little bit more and more because of the mechanical function that you want to have, and the, let's say, approaches where you measure bone density. They are not really so prone that you can. How should I put this? I mean, there you don't have a mechanical function in it, right? They are just showing you a bone density, and that by itself is an important predictor of the stability of the structure, but it really doesn't help you to understand the mechanics. You're not measuring, or you're not imaging mechanics. And that was one of the things that we know. Of course, mechanics is not on one side, only the stability of the bone, but also it can adapt, the bone can adapt, right, to this mechanical function. So age-related bone loss has been attributed to the decrease in mechanical usage of the skeleton. This is also an important part. As we get older, we're probably slightly less active, or if we are active, our body doesn't respond anymore as much to these mechanical cues. So on the other side, if you can load mechanically, this results in enhanced bone formation. And so for us, a detailed understanding of the mechanobiological, not just the biomechanical process governing load-related bone remodeling down to single cell level will really help identify also new molecular targets for prevention and treatment of bone fracture, which is the area that we're more and more going into, right? So we are often using antibodies, for example, for treating and biological assays to treat not just chemical pharmaceuticals, right? So they are much more biological. So you have to understand the biology, and that kind of fascinated me. And that's why I moved a little bit more into this mechanobiology field, using very similar tools though. And I want to guide you from how we came from structural imaging to functional imaging to going into more mechanobiological imaging and combining different modalities. And that's the tool we're going to take together. And maybe just on the side, it's very important. This is a lot based on some ideas that Julius Wolff postulated, in written, in how bone adapts to mechanical load. But there's also a direct connection to ETH, where I'm, of course, currently a professor, because these figures that you see on this side here, they would actually allow you to calculate the main stress fields. And this is this is from Kuhlmann, and a professor, an early professor in civil engineering at ETH, who invented that way of showing actually these kinds of stress trajectories. And so, of course, Wolf has then shown that this aligns pretty well with, for example, the architecture here in the femur. And so here is a pair of this same type of images with a normal bone on the left and the osteoporotic bone on the right. But now you don't have to imagine anymore which one is stronger and how they fail, right? You see the effects of osteoporosis much clearer because we have used the mechanical testing in combination with the micro CT images. So this is so-called a stepwise microcompression. So what you do is you put a 8mm diameter biopsy into 2 platens, and then you do basically step by step by step by step. You compress, and you can see now much more the function, right, and how it's regulated. And it really helped us a lot to understand what's going on. And you can, of course, also then quantify these kinds of things, and you will see that, for example, in the normal bone or healthy bone on this side, you get like the weakest link of the chain approach, where there's a little bit less bone in this area, and that's where the bone structure fails. Whereas in the osteoporotic bone, you have catastrophic failure in multiple places. And this is important that first of all, it's more difficult to predict where it's going to happen, but it also is not just, nature has built in these areas where it's not so dangerous to fracture. If you have to fracture there because maybe there's not a growth plate around, right? So if you fracture in a growth plate, that's really bad. So if you're an elderly person, the fracture will happen maybe at places where it's much more difficult for the cells then to fix it, right? And besides the fact that they have less ability to actually fix it because of advanced age of the person, for example. So what you can do, of course, with image processes is then also to identify individual elements. And what you will see is that small, tiny little trabeculae, they have a high tendency to buckle and fail. And then also the larger structures like these plates, they normally don't fail. But if they have, like here, a fenestration happening, that's a typical hallmark of aging, then of course the fracture actually happens around this place. And that's something that is shown here with a higher resolution technique, synchrotron radiation-based micro CT. And this is not just the higher resolution that's important, it's also a monoenergetic beam, means that you have much better visibility of these cracks that are developing. And so that's what you see here. You can see very nicely that it's on these convex structures, like on the outside here. Yes, there is a crack forming, but most of the cracks are forming from the other convex structure, which is this hole inside. So with making holes in the bone, you're increasing a lot the convexity of the structure with this higher chances of fracture. And so this is a very important process that we need to understand and help us really, say like sometimes, for example, that some of the drugs might have a huge effect with only a little bit of bone formed. Let's say if you can fill this hole again, then your likelihood of fracturing is much reduced because this will not happen. It will not initiate failure. So when you look at that, then it is high resolution. The nice other part about imaging in 3D and having potential ways of image processing and really have a binarized thresholded images. You can, of course, for example, take all away these osteocytes that you see here in yellow, and then focus on these canals here that you see. They're mostly blood vessels, but they could also be just resorption canals, and so void space basically. But they're very important because these void spaces, they are again, this is in cortical bone now rather than in trabecular bone. They're again the stress razors that you see in green, all of this, cracks. So a crack is not a linear line. It looks like in 2D like that, right? But it's really a wake plane. And you can see all kinds of features. It looks like we don't have enough resolution, but they're actually very important, these little holes inside, because that's where you have like ligament bridging between the materials. There's still collagen, so we can't measure that in a micro CT, but this keeping everything together, and this will expand, and we also have of course, visualization possibilities to show like how cracks develop and go over time. And I show you a little bit more on that part as well, because I think it's quite important and will guide you towards this more mechanobiological part, right? So we have all of these things that are happening. So we were wondering, so what about the osteocytes, right? It's not so clear in the image how they contribute. And then now you have the image here that is animating, and you can see here is a big hole. It's coming up. And from that hole, you have a crack emanating. Actually, you can see here, right here, right? So this crack is coming from this hole. And then it, grows outside, right? Like this way. And if you follow that path, you see like the osteocytes are in the way. Like the cracks go through the osteocyte lacuna, but they are following also. They're not just always between the osteocytes, right? But they are following the osteocyte lacuna. So that means that, yeah, they contribute not to the initiation of the crack. So a bone will never fail because there's an osteocyte lacuna or too many or whatever shape it has, but the number and, will guide the crack where it goes through. So this makes something relatively special because then also the, osteocytes that are sitting there will be always feeling, right, these kinds of crack happening. And there's of course something they want to avoid, because if the crack goes through them, they're gonna die. Okay, so this is goes in apoptosis, this cell, and then some remodeling processes are turned on because of this cracking. This cracking actually doesn't need to be necessarily catastrophic all the time. So you have microcracks, right now, all of you have microcracks in your bone. This is a normal process and helps actually to renew bone. And so, whenever you probably jump from a little bit too high height or went jogging a little bit too extensive, then maybe you have developed some microcracks. And they are healed, right? And within, let's say, 3 years or so time frames, right? This is the normal turnover in your bone. So this is an important signal that you have that shows that bone should be remodeled. So they are important. So that first time, this is quite some time ago, that we did this kind of work. You can see already starting in 2001 and then 2009, some important papers there that show that, okay, we thought, oh gosh, we were just interested in the bone and the failure, but hey, these osteocytes, they're probably going to be quite important. Okay, so jump out a little bit of that. This is all experimental work. It was highly nicely animated, but this is all coming from real experiments. But, remember the images that I showed you, how bone is failing. What we also did, like, we cannot always go in there and fracture the bone, because otherwise, again, the osteocytes are going to be dead. What about if you want to know what environment the osteocytes have and what kind of mechanical environment? We need computational methods that allow us to compute that. And it's just an example to show here that we can really nicely emulate these failure processes that I showed you by using computational tools, right? But we don't know it to go there. This is a nonlinear analysis. You cannot get real deformations, of course, in nonlinear analysis. But the osteocytes, they work in a linear phase when bone is not fracturing all the time, right? So it's very important that we can use simpler methods even to define the strain field, because it's a strain field that is driving these biological processes. I just wanted to show that, but then we can also use these calculations of the strain field, for example, to simulate, in this case here, this cracking mechanism. So this is just showing you, right? You see a crack growing from this, this is really a simulation, or quite demanding simulation, needs a lot of computer time. And you can see the normal gradient model that we use. It's just, basically the only important thing is this crack here, and the mechanics is all defined by the crack. Then if you do this so-called scissor model, where you assume that all the osteocytes are connected with each other by the lacuna canonical network, you can see that now it's actually the cracks following those osteocytes as well. And as we saw before in the experimental data. So this is quite an important thing, that somehow, it's not enough. If you just take the osteocyte lacuna as a increasing stress razor, it is, but that's not enough to do something with this high energy crack that's coming in. But if you take the other volume, that is basically this it's about the same porosity that you have in osteocyte lacuna than in osteocyte dendritic processes, right? Or like the canalicular processes. It's about 1% of the matrix. So, both. And so if you do that, then you understand better how cracks actually develop. This, okay? A little bit basic science, I'd say, in terms of the mechanics of it, but quite fascinating. And okay, again, tells you that, okay, if you believe that there's a cell inside that can sense the mechanics, this is a very important part, right? Because it's constantly feeling that through the lacuna canalicular network, and not just because it has, directly cracks coming to them or mechanical forces directly coming. So the exchange in the lacuna canalicular network is extremely important. So now we have also, in terms of the imaging, we can of course get higher and higher resolution. So these are some images that were produced by Elliot Goffin. This is a PhD and published, last year. And this is where you can see how you can really assess millions of osteocytes now in a piece of bone at 1 micron resolution. And you can see they have come with all kinds of shapes and forms, these osteocytes. And that's actually quite important. The biggest difference you can see is between cortical bone and trabecular bone. So that's a big difference. That's what you see, the cortical bone up here and the trabecular bone down here. But if you look at, for example, here in between, this is 100 samples that we measured in this kind of really high resolution. And you can see, like with idiopathic osteoporosis, or with low density, you don't actually see differences between the groups. So it is something that in the disease states, this is probably not a very good predictor, right? In that sense, osteocyte lacuna seems to be a high variation in what it looks like, but they are specific for the tissue and for the site but they are uniform between people. Okay, so this is quite an interesting fact that the cellular function must be quite uniform. If you have a problem though with the cellular function, then you see the effect very quickly because it's quite radical and dramatic. And that's something I just want to have you take with you. And this is not another link. There is a direct link to mechanosensitivity. So the shape of the osteocyte and how spherical it is, so sphericity is like how sensitive it is. You can see an oblate structure with a very low sphericity that we basically 10 times higher in terms of mechanosensitivity or the stresses that are built around the cell than one that is perfectly sphere or even in a prolate structure, which you are protected almost right from that. So there you don't feel much anymore. But let's say this is what you find in humans. So this is the sphericity. You can see it's tightly controlled. Although you had a good impression of the variety of cells that I showed you before, the sphericity of them are not very much changing, right? You go from 0.27 to 0.78 or something like that, very small changes. But look at how this is directly correlated with the bone density that you have, right? So what it tells you as well, if you have very low bone density, you have more spherical osteocytes, means they're less mechanosensitive. Okay, so they're less mechanosensitive, they feel like the environment is not mechanically loaded, means you don't need a lot of bone there, right? If you have very high bone density, then the osteocytes are optimized for that. So they feel much more the mechanics that are going around and say, okay, now you need more bone there. So it's hard to tell what's the hen and the egg in that sense, right? Whether one is driving this or the other. And I will not be able to show you today because we don't know, which is the process. So is osteocytes adapting to the high density or is it driving the high density? We will not know that for now because this is not possible to measure that time lapse. And so we can only do endpoints right now with this kind of high resolution imaging. So we never know which one is which. But when we look at mechanobiology at the cellular level, it's important that we have maintenance and adaptation of bone morphology, which results from this orchestrated remodeling process, right? So really built the stage that we not only want to look at, the stability of these structures, but really like how do they adapt to mechanical loads. And this is locally coordinated by these osteocytes, another beautiful illustration in how they are in such a bone structure, and they have used biochemical signals. They don't use mechanics, of course, to translate this information, but of course the biochemical signals are produced by some kind of mechanical stimulus that is there, or some of it are produced, and we're interested in that. And this will result in increased or decreased bone formation or resorption activities. And so to better understand bone adaptation, we therefore have to understand how osteocytes sense and process mechanical loading within their local microenvironment. And you can see here very nicely, these are two osteocytes, so this is half of osteocyte, this is the full osteocyte, and these are the dendrites that are connecting actually these two osteocytes. And these is not now done with micro-CT, this is a FIB scanning electron microscopy image that you have 30 nanometers stack or slices where you go through, and you basically use a focused ion beam to evaporate, say, layer by layer, right? But you get beautiful instructions here in what these look like. So if you want to do these kinds of things, very hard to do it in human because of a lot of variation, and we don't know much about that variation. So people have decided that mouse models might be of interest in that sense, because we have an inbred strain that is 90.9999% genetically identical with each other. So you don't have to deal with genetics, but then you can use different animal models with different genetics to compare, for example, the effects of genes you can knock in, knock out, and all of that. You're probably aware of that. But for us, it's very important that the procedures are reproducible, right? So that we need a reproducible procedure, what we do with these animals. And here, for example, this model of bone adaptation is using the tailbone. We have 3 tailbones here. They are loaded between the 2 platens. And you can see that here, with 10 Hertz, 3000 cycles, we do that 3 times a week for 4 weeks. So 5 minutes of mechanotherapy. That's meant to be a therapy. It's just 5 minutes for 3 days, right, per week, and then we do that for 4 weeks. And when you image this bone with micro-CT over time, this is what you see. And you can see very nicely how you go from this structure to have a structure that is now thickening quite clearly, and you have more bone density. If you quantify that, you can see that you go from 100% here to about 120%. So you're gaining about 15 to 20 over the normal development of this mouse, that's 15 to 20% more bone mass by just 5 minutes of exercise, 3 times a week. Okay, so that if you could do that in humans, it would be fantastic. It's not so easy, right? Because one of the things that's important to know is that you use very high forces. So this is 8 newtons. Not a lot, right? So if you think about that, that's like 800 grams that you would put on weight. So it's not a lot of force, but for that bone is half of the breaking strength. So it means that if you want to use that in patients, you would need to go to half of the breaking strength of a bone. If you don't know exactly what the breaking strength is of the bone, I'd be careful with that, right? Meaning that you might be, damaging the person's bone, right? With mouse, we know what the breaking strength is. They are genetically identical. We know that they never fail, totally fine. But in humans, it's not so easy to translate that. Okay, again, take another journey. And for the ones, maybe some people have already seen that. This is going inside some of these vertebrae. What we've done using finite element analysis to predict the local stresses and strains. And what you see here is in red is where bone is loaded highly or maybe even overloaded, but that is not clear. This doesn't matter. It's just that there's a high load there, high strain. And where it's blue, you can see areas where bone is basically lowly loaded. And so if you then now go back to Wolff's law, it would tell you that bone should be formed where bone is highly loaded, and bone should be resorbed where bone is lowly loaded. So now you can see in the trabecular structure of this vertebra, we have a huge playing field because you have all kinds of scenarios, right? So you have not just one. If you do it people do a lot of cortical bone work on this, but this is much more difficult because the mechanical prediction that you do is much more homogeneous. And so not much changes, but here you have huge changes over time, and we can make use of that. And if you do that, this is some older work. 10 years ago, Friederike Schulte has published a paper where she was able to show that, if you look, right, this is an area of initially, before we did mechanical loading, was quite highly loaded in the normal scenario, physiological loading. And then if you do looking at experimental data with time-lapse micro-CTs, you have 2 pictures, and you basically calculate where bone was formed and where bone was resorbed. Bone was formed in the orange, bone was resorbed in the this part. You can see, yes, bone was actually formed here. And you can also see that this is an unloaded area or lowly loaded area. And then you can see this bone has been resorbed. Okay, so, and then of course we need to show that quantitatively, and we can show quite nicely and quantitatively that this is actually true for, on average everywhere, right? So we can show that, yes, low strains are associated with bone resorption, high strains are associated with bone formation, and there is mechanoregulation. And this mechanoregulation is available physiological state, mechanically loaded state, treatment, overrectomy, everywhere. It's always there. It's never going to go away. You never have this fully dysregulated, mechanics. That's never happening, because the mechanics, if the mechanics doesn't work anymore, if the cells are not responding to mechanics, then some really weird things would happen. But we have not found yet such an indication where it's totally dysregulated. But it's just alert, and with also the consortium that we're working out, and where I know Leila from Fidelio, where we're interested in diabetes, this might be such an implication where maybe there is some dysregulation of mechanics. Okay, so what happens if you have a fracture because you had that problem? We can also do time-lapse imaging of fracture healing. And so what we do here is we use this external fixture, we put it on the femur of a mouse, and we use mechanical loading again here. That is, basically the 10 hertz again, and 3 times a week. And we see how this affects actually fracture healing. And this is what is shown here. So you have the loaded in week 1 and week 2, there is no loading. And then when you start with week 4, you also see the effect of loading, because of course, first you have to have some bridging that the load can be transferred. And you can see very nicely how you get a much bigger callus, much more stable bone than in the control, for example, right? So loading also in fracture healing is an important part. And then you can also associate areas, again, of high mechanical forces and intermediate and low mechanical forces, where you have bone formation happening in high strain areas and you have bone resorption happening in low strain areas. So the same mechanism in fracture healing happening than what I showed you before in bone adaptation. And then we can go further, because what we often do at the end of a study, we are creating histological sections in the classical sense of bone morphometry. And what Francisco did was to basically register histological sections, take out here the cells that are stained for sclerostin, piezo-1, YAS, TAB, all kinds of rank ligand, and you co-register with the images. Okay, so it's a very important process that you can bring this information into it and have a multimodal approach where you correlate these different findings, but all in one package. So there's a lot of, again, information technology inside that allows you to do that. So when I then have special mice, for example, here we have some mice that have they are prematurely aging, and they have been engineered with CRISPR-Cas9 to produce fluorescent reporters for formation and resorption. You can link again this all up, right? So we see experimental data where bone forming cells are and bone resorbing cells are. And if I look at my micro CT image, I can link this up actually and see like, okay, there's bone resorption here. Yes, there's osteoclasts here that actually do it. There's bone formation here. Yes, there are osteoblasts here that are actually doing this. And then I can see that this is also linked to some of the mechanical strain maps that we do. So the same thing is really, really intricate information on biology, mechanics, and the mineral that is formed. And I think this is what really a specialty today. We can use in silico predictions of that. I will fly through that a little bit faster. And, you know, because you don't always do manual work, so you want to try to replace and reduce certainly some of the manual work. And we use some simulations to actually do that. So where we have the basis of the images made from micro CT images in vivo, and then we can do simulations of all kinds of cells and physical properties in the matrix, and then predict cellular processes and how they build bone. And I just want to show you that, for example, what it would look like. You just saw before in vivo data and how this looks like. We even go a little bit further. Jack Kendall here has really produced, you can see the vessels are always first. They are growing into these matrices. And then you see how the bone is actually forming around that. Very similar appearance as what you showed or what was shown in vivo. And he has since validated this approach and really gets very similar results, quantification, that we can use maybe such models to predict better in the future the effects and look, for example, how, because we have cellular information, they have gene expression, protein expression, and we can, for example, interfere with their parts and look at that. But what kind of proteins or what kind of genes are actually expressed when you do that? So I go back to our loading experiment, and we have developed a technique which we frame local in vivo environment imaging, because we have this enriched environment, right, that we want to do. So we do 2 in vivo micro CT images. We can do a bone remodeling map. We can do a strain energy density map. So it's time-lapse in that sense of as you go with time. Then we go to the end of the study. We take a histological section. In that section, for example, in this small region, we have all of these osteocytes inside. Then we use, for example, here, laser capture microdissection, or we use some other more modern spatial biology techniques like Visium from 10x, so you can use nanostring type of approaches that allow you to look, you know, at this cell in more detail. But here we really basically extracted these cells from the section, and that's what it looks like. You have a laser, and the laser goes in and does a circle around the cell. The cell is sitting inside here in the section, goes around 3 times. Once it has been cutting through the section, it uses a laser pulse to catapult out this in an Eppendorf tube. Right, so this is going in an Eppendorf tube, and show that just a second. But what's very important, I have now information, spatial information. I know this cell, exactly where it came from. I know what kind of forces and what kind of remodeling maps are associated with each cell. I can have hundreds of cells, could theoretically have thousands of cells, but that's really time consuming, so we can't do that. I think the biggest we ever did was 170 cells. It's already quite a bit. But that's very important. So we have all of this information, and then we can send this on actually to do quantitative PCR or RNA sequencing even, and get information out of that. And I just want to show you one result section. So this is for 3 genes, SOST, OPG, and rank ligand. I don't want to go into detail in that, but SOST, of course, is coding for sclerostin, which is a human antibody available that is used in the clinics, right, to do that. Rank ligand is the same. So anti-rank ligand, for example, is a drug, denosumab it's called, that allows you to have antibodies. So we choose some of these genes that are coding for proteins that are important and used in the clinics. And so it's quite important when you look at that, right? So when you look at the expression level in SOST, you know, right, if you have high, would say that if you have high loading, it should be reduced. And you can see that to some extent, right? But it's not so clear. It's also not significant. OPG is not doesn't matter whether the cell is coming from a highly strained area or lowly strained area, but rank ligand, right, which is basically signaling the osteoclasts that they should come in and resorb bone, is not available in areas of high loading. There is never an osteoclast seen then in that area. Intermediate, it's reduced, and then normal is in areas of low loading. So that really is really protecting this area, right? The loading makes sure that if you have high strains, then no osteoclasts go close then. So if you use now, what happens if you use additional loading? What you would also expect is you can overall SOS expression is downregulated, but not in a site-specific manner, just overall. OPG starts maybe also be a little bit regulated, but here, you know, rank ligand again, right? Basically, if you do extra loading, you don't give osteoclasts a chance to work. There is only in very loaded areas that there is rank ligand expressed and 10 times lower in the expression, so not much should happen. Okay, how you want to illustrate that? It's not so easy, right? I go very fast, of course, but it's very important that you can gain a lot when you go from 2D to 3D. And I just want to show this. This is now for sclerostin. You can see this, of course, you know, what you see is a sclerostin positive is black and sclerostin negative cell. And that has come from histological section that was linked to a micro CT. And you can see the remodeling map in 2D and the SED map. But, you know, formation is here, resorption is here. It's not much you can say about these cells, right? Because you don't see the full context. What you have to do is to really go in there to do it three-dimensional. And then if you do that, you can see, of course, that there's an association with areas of resorption, formation, but also very important in areas of high and low strains. And so this is an important part that in 3D, you have the context and you can also look at how you have changes because you have time lapse in STD, which seems to be even more important, that you're going from highly loaded to lowly loaded areas over time. That predicts somehow whether you have more or less sclerostin expression. Just to stop with, I want to show this beautiful animation here that shows you the different states that you can measure from micro CT, formation resorption map, FV, but you can also see how now we have registered all of the cellular information, and hopefully we could do that for the full 3D extent, where you can see where they're located, and you can start making your stories, right? And how is this associated? Why are the cells expressing a certain protein? Why are these not expressing? And so you have such a rich environment to study, which is also difficult, of course, to go through. It's a lot of data, and we have to learn how to interpret such parts. So, in conclusion, I showed you that hierarchical imaging of bone facilitates better understanding of osteoporotic fractures and prevention. I've also showed you that time-lapse in vivo imaging, but also time-lapse biomechanical imaging allows this longitudinal quantification of here bone adaptation and regeneration using multiscale mechanobiology approach. And this live imaging that I was talking about allowed clarification of the role of a wide range of genes. So we can really do RNA sequencing, and so we get basically the full genome information and see how it's associated with these different states as key players in the mechanically induced bone adaptation and regeneration process. Spatial mechanobiology also informs cell-based in silico models that I showed you for the fracture healing, and here's one shown for bone adaptation that allows you to predict realistic outcomes for bone adaptation and regeneration. So it's hard to see, but you can see here how the cells actually moving around on the structure and how they remodeling the structure over time. And in the future, this hopefully will facilitate better understanding of biochemical signaling pathways in bone adaptation and regeneration in vivo. And with this, I'm already at the end, and I would like to thank, of course, the team. This is a picture from last year. It's already changed again. We have about 30 people now in the lab, very diverse backgrounds from chemistry to physics to engineering to biology to veterinarian sciences. So I think it's very diverse. So if you're interested in looking into that, maybe your background fits also quite nicely. And the funding agencies, of course, have been involved specifically for the things that I've shown. So there's, of course, a very important part, but also like to thank really ETH, who is providing a lot of this backbone for us. So it's really a university that invests into research for their for their investigators, and so that I'm very thankful for that, to be able to get that kind of funding actually. And with that, I'd like to thank you for your attention, and I'm open for questions.